So I'm going to talk about penguins. They're nice. This is actually a thermodynamic journey. I'm obsessed. I mean, I'm a biochemist, so for me, everything is biochemistry. Thermodynamic journey of a penguin colony. I was actually watching a movie with my kids, fantastic documentary about penguins, March of the Penguins, when I suddenly realized that penguins actually happen to live as in different isoforms. They have pups and then they get old. They clusterize. They can actually form small oligomers of penguins and even get together to form very ordinated and very large clusters of penguins, aggregates actually. And why they do that? They do that to survive the storms. In fact, these large aggregates of penguins are actually making the penguins quite resistant to the strong cold occurring in the Arctic. The next slide screw me up the thing. Of course, I'm going to talk about prions again. This is a thermodynamic journey of a polypeptide, though, and it's surprisingly similar to the penguins. We start with a pup, which is actually just a polypeptide chain, which, of course, acquire a conformation. In the case of prion diseases, we call it PrPC, of the cellular form of the prion protein. This is the conformation that allows the protein to exert its function. And then in pathological conditions, we know that it's actually not black and white. We don't have PrPC and PrPSC. We have PrPC and a variety of misfolded isoforms, which we are still trying to define. PrPSC is probably just the end point of it. And like the penguins, if you remember the first slide, this is the one easiest to detect and easiest to retrieve in the brain tissues because it's very hard to break it down and it's very resistant to treatments. But we really don't know which isoform is actually not only responsible for the transmission of prions, but also responsible for killing the neurons, and we're still trying to define that. Now, this sort of pathway is now, we know, is common to several diseases. We have the example of the A-beta plaques, which characterize Alzheimer's disease, Lewy bodies linked to Parkinson, tauopathies, where the protein tau gets highly phosphorylated and forms neurofibrillary tangles. And then, of course, our PrP, scrapie, and prion diseases. Now, what's really the peculiarity of prion diseases is that we believe this has to do, is actually an example of transmission in absence of nucleic acid. So PrP, scrapie get in touch with PrPC, and after that, PrPC becomes PrP, scrapie, and after that we have basically an increasing amount of the infectious agent PrP, scrapie. So this has been debated for years. Dr. Prusiner finally got a Nobel Prize at the end of the 90s, but actually after probably 20 years of fighting with the rest of the scientific community, thanks to mammalian prions, but even more to prions in other species, in particular in the yeast, we now, I guess, most of us believe that a protein can be infectious, and so the infectivity of PrP, scrapie is well accepted now. What has been given as a normal kind of logic next step was that PrP, scrapie accumulate in the brain and then damage it, and that's why actually we have degeneration. I can say that I'm pretty confident in saying that that PrP, scrapie is actually not the only cause of the degeneration. We now know that very well. Famous cases of toxicity, especially in experimental models in lab, toxicity in absence of infectivity, the opposite, infectivity in absence of toxicity. And what has been shown is that there is something more than just PrP, scrapie required to kill the neurons. What we know is that actually the normal form of the prion protein, the cellular prion protein, the benign form, the one that normally is expressed everywhere, is a crucial part of the game here. So we have probably, this is just a model, but it's recapitulating years of studies. PrP, scrapie get in touch with PrPC in his attempt to recruit the protein again to induce the misfolding, but something happened in this particular moment, because then PrPC is required to transduce the toxicity probably coming from some isoform, some misfolded isoform of PrP scrapie. So we need PrPC on the surface of the neurons in order to have toxicity. Just to give you a very brief example, if we have PrP scrapie and we deplete PrPC from the surface of the neurons, neurons don't die. Or eventually they die very late after massive amount of PrP scrapie accumulated. In that case, it may be a mechanical thing, but probably that's not what happened in the real disease. So PrPC is part of the game. That's the important thing, because now if we go back to the pathway of the penguins, we have, let's pose this question. What is the best pharmacological target in prion diseases? We have the polypeptide chain, which of course is the precursor of everything. This is well known. If we deplete PrPC, actually the gene of PrP from an organism, that organism becomes 100% resistant to prion diseases. That would be ideal. Practically speaking, is not easy to do these days. We hope in the future will be possible more easily. Then we have PrPC, the native conformation. I show you that it not only acts as a substrate for prion replication, it also acts as a kind of modulator, transducer of toxicity arising from some isoform of PrP scrapie. Now, PrP scrapie, of course, is one of the targets. The difficult thing, and in fact, actually, in the last 20 years, people have been focusing on drugs that can target PrP scrapie, the famous high-throughput screening done by the Prusiner's lab that eventually came out with very interesting compounds. Unfortunately, there are two main caveats in targeting PrP scrapie, which are emerging. It's not just my opinion. The first one is that we don't know what we're targeting. Because as I explained before, PrP scrapie is not just one isoform, for which we don't know exactly the structure, but it's probably a variety of isoforms. And related to that, we know about now very well about the prion strain phenomenon. So in fact, some molecules that are very active against one strain of prion are completely inactive against another one. So, and it's, I mean, conceptually, it's becoming more difficult to target something that is so elusive and difficult to catch. So my lab has decided to actually focus on something that we know very well, which is the cellular form of the prion protein. For this protein, there are several advantages of deciding to go with a drug screening against the cellular prion protein. First of all, actually missing a slide. Well, anyway, first of all, we know the structure of PrPC very well by different biophysical methods. We define exactly the conformation. We then can apply very rational kind of approaches to target it. And on top of that, PrPC, as I showed you before, it's the beginning of everything here. So think about prion strains. Whatever strain will need the substrate to propagate. So let's say if we are able to block the substrate, ideally, theoretically, no matter what strain we're talking about, will be blocked, because we block this sort of reaction at the very beginning. These are kind of the concept that we are applying here. So we decided to target, we are actually currently running research programs to target PrPC and of course having prion models in cells and mice as a readout. What we're trying to do are basically three approaches. We try to remove PrPC from the surface so that the infectious agent PrP scrapie doesn't find its substrate, and eventually the transducer, so PrPC itself, is not in place, in the wrong place in this case, because no PrPC on the surface, no disease. We know that in many models. The second approach, which is the one that I'm going to present today, is the idea of pharmacological chaperones, and I'll talk about that in a second. And the third one is actually to inhibit whatever downstream pathway elicited by PrPC after getting contact with some aberrant isoform, misfolded isoform of PrP scrapie. So for the rest of this talk, I will talk about this one, pharmacological chaperones. And I'll try to give you just a very brief explanation of what is the idea behind it. Again, we have the polypeptide chain that assume a conformation. This is actually now a thermodynamic graph. So energy, it's in this way, it's in this axis. The native conformation definitely has less energy than the starting polypeptide. Somehow, driven by mutations inside the polypeptide, or in a stochastic event, for the case of sporadic CJD, we believe that there is a kind of an energy barrier that the normal conformation of PRPC pass, and then forming some misfolded conformation, the one that I was referring before, still kind of uncharacterized. The endpoint, we know it very well, is PRPSC, which is the lowest energy possible here. It's very resistant to treatment because it's very stable. So you might think that breaking this up or breaking this up may actually form some even more toxic species, perhaps. Definitely increasing, breaking up PRP scrapie will probably increase infectivity. And going back, it will have to mess with a very strong energy barrier, perhaps even impossible to do. So we decided to go with this idea of applying the law of thermodynamics in order to decrease the energy barrier that is required for PRPC to become something misfolded. If we are able to attach a small molecule that is strong enough to interact with PRPC, then thermodynamics tells us that we're going to lower the thermodynamic state of the substrate, in this case of PRPC, and increase the energy barrier. So this favors the entire process. Now, before we started, of course, we asked to ourselves, are there compounds selectively targeting PRPC already available? To my surprise, when I started this project, I found that yes, there were many compounds in the literature claim to be ligands of the cellular prion protein. So we decided to test them all by using just a couple of techniques. I'm not going to spend too much time. This is a biophysical technique called surface plasmon resonance, where PRPC is attached onto the surface of a chip, and then we can flow through small molecules and see binding. Or a very simple and old-fashioned technique, which is equilibrium dialysis. This is just basically, it has to do with the principle of putting a membrane that is permeable to a small molecule, but not the protein, so we can see the equilibration of the small molecules between the two chambers, one without the protein and one with the protein. So we use these two different techniques to then test the hypothesis that some of the molecules claimed in the literature to be PRPC specific were actually really that. To our surprise, though, we found some compounds that were kind of really binding to PRPC, but in a kind of non-specific fashion, meaning that we're probably binding to PRPC on top of several other proteins or cell components. And this is actually true for the polyanionic polymers and some porphyrin, the iron tetrapyrrole published by the Collins lab, which is actually a very good antiprion compound, but again, it has poor specificity for PRPC. It has many targets. Other compounds, actually, we were not able to reproduce the original data. I'm showing just one. This GN8 is a compound published by this Japanese group as an antiprion compound and as a ligand of the PRPC. So this was actually seemed to be an example of a pharmacological chaperone, exactly in the way that we were looking for. Unfortunately for us, we found that the GN8 is not binding either by SPR or equilibrium dialysis, although we confirmed that it's a very potent antiprion compound. In cells, it has an IC50, an inhibitory concentration 50, similar to quinacrine. Our data are actually saying that most probably this compound is not acting through PRPC. So then, of course, we decided to embark in attempts to find our own ligands for PRPC. And again, taking advantage of the known structures of PRPC, we ran this campaign of in silico drug screen. So basically, we started by the available information and ran computer algorithms to try to define first pockets on the surface of PRPC that were possibly hosting the binding of small molecules, and then rank these sites, and then started to really screen molecules inside it. And then, of course, the computer made predictions, and we had to confirm the predictions in vitro. This is a general schematic of the algorithm, of the entire algorithm that we built. Surface plasmon resonance was our primary screen. We actually came out with these 4 molecules. DS is for drug screening. We're not fancy giving names in this case. Among these four small molecules that apparently were able to bind PRPC, we picked the last one, DS26, which seemed to be binding at a very interesting affinity as a starting compound. In fact, its affinity for PRPC was actually sub-micromolar. And then, of course, we started characterizing the S26 from a biological perspective. We found that the S26 is an anti-prion compound. It actually works pretty well. And we were able actually now to define its activity against two different prion strains, RML and 22L. One thing that came into our attention very soon is that the S26 was binding to PRPC at a submicromolar concentration, but we needed very high amount of the compound in order to be effective. In this case, 100, 300, and 500 micromolar, so 3 orders of magnitudes higher concentration than its expected KD. And to make a long story short, what we suddenly realized was that the pharmacokinetic properties of the compound were very poor, meaning that the compound was basically not surviving long enough in biological fluids to be really active, not including the medium for the cells. That's why we needed so much. The compound was actually not able to penetrate the blood-brain barrier, which prevented us actually to test the compound in vivo. And on top of that, one of the reviewers of a paper that we sent out last year pointed out that there was something wrong with the kinetics. He was totally right. Sometimes actually reviewers are very good. We discovered that the S26 actually has a very kind of unexpected behavior. It may form clusters, aggregates itself, and as an aggregate then, it may bind also to other regions of the prion protein, not just the one that we predicted in the C-terminus. So to make a long story short, we try very hard to modify the chemical structure of the S26, and this was inconclusive. It seems that there was a strong relationship between the chemical part of the molecule that was really giving us the results on PRPC binding and activity, and the poor pharmacokinetics, the blood-brain barrier penetration, and the aggregation. So basically, the S26 is very hard to modify. Anytime that we try to modify it, we lost the activity. So what we're trying to do now is capitalize on this information. We know that we can accomplish it. So we're using recent technologies, first focusing on the chemistry. We learned that we have to give priority to chemistry, or we might end up having a dead road, like in the case of the S26. And on top of that, we're also applying more recent techniques like this label-free from PerkinElmer, which allow us to increase the number of samples that we can manage. This machine allows us to test binding of 384 small molecules at a time. On top of that, we're actually rationalizing the other binding pockets that we found in the previous screen and running, again, virtual screenings on that. One of the first candidates that emerged from this sort of attempts is this molecule called SM72. SM72 actually has a KD not that bad for PRPC, but much higher than the S26. So its affinity for the target is not outstanding, but still is not bad. The most important thing is that SM72 works against prion-infected cells against prion replication, and this time the concentrations are closer to its predicted affinity, actually calculated affinity for PRPC. We recently also observed that the compound works also in the protein misfolding cyclic amplification that Christina showed before. Again, but probably the most important thing is that this is a drug-like. This is a scaffold that we, on top of all the molecules that we selected before, is suitable for optimization. So it's not like the S26 before. And that's what we're doing these days. We're testing hundreds of different derivatives of SM72 with the purpose of, one, lowering actually the KD for PRPC, so increasing the affinity. Two, look at the biological effects. And this is actually a schematic of what we are really doing. We're taking into account the pharmacokinetics of the compound in order to be able to get in vivo through chemical optimization and reevaluation of the biological properties of the molecules. Our objective, as far as now, is to get a molecule that can pass the blood-brain barrier and we can test in animal models. This was a slide that was supposed to show up before. Conclusions. PRPC to us represents a very convenient, actually the most convenient pharmacological target in prion diseases. We now have proof of principle, at least, that it can be targeted and show basically good properties against prions in cells at least, definitely provide an unprecedented opportunity to overcome the problems that affected previous screens, in particular prion strains. We are currently pursuing different strategies to target PRPC, and we have few leads that we're trying to optimize. Now, everything that I presented has been done by these three ladies, Claudia, Tanya, and Sayoa, and a wide network of collaborations around the world. And also thanks to the Telethon Foundation, you, of course, with your support, the Fondazione Veronesi, the Italian Ministry of Health, and the eRare project of the European Community. Thank you for your attention.